Hi, I am Revati, Assistant Professor, Department of EEE, SNS College of Technology. In this lecture series video of Biology for Engineers, today's topic is enzymes. So this is the important topic in biology for engineers, whereas enzymes plays a major role in your body growth and all the living organisms. So enzymes are biological molecules, mostly a type of proteins. They speed up chemical reaction in living organisms. Without enzymes, most reaction in body would be too slow to support life. So it supports the quick response in your body, and it speed up the chemical reaction, whereas chemical reactions plays a major role in your digestive system, or else in circulatory system, or else in the excretory system, where else it's included the lipid protein digestion enzymes releasing. So everything comes under these enzymes, which when the body is lagging in enzymes, it's takes the process of body which supports the life is too slow. So properties of enzymes are highly specific. Each enzyme works on a particular substrate. So it contains a substrates, whereas it's a group of components where one types of enzymes works on the particular substrate. For else in digestive system, or else in an excretory system, or else in circulatory system, it works on a group which is in a particular area. So reusable. It consumed in a reaction. Works best at optimum temperature and pH value. So whereas in pH value, we all know that acidic and base value of a substance which is present in every chemical substance in your body. So works based on optimum temperature, which is also considered as a room temperature or else your body temperature. Speed of reactions by lowering activation energy. So activation energy is a type of energy which may activate your inner parts of your body, which is speed up by the enzymes. Structures of enzymes are made up of long chains of amino acids folded into a unique shape. The active site is where the substrate binds. So the binding of substance, which is also known as an active site. So enzyme substrate complex forms during the reaction. So already I said that enzyme works on a particular substrate. So the combination of enzyme and substrate complex forms during a reaction. So in a chemical reaction, whereas the body which is responsible for the chemical reaction, the enzymes and substrates combines together and form a complex form. So enzymes acts in a substrate maltose, which is combined with the enzymes. The combination of these two is also known as an active site. So the releasing of a chemical reaction, which is called as a glucose. So how these enzymes work? So combination of these substrate and enzymes forms a chemical reaction, whereas how it is working. So the lock and key model under the induced fit model. So these are the two types of enzymes models which works on the principles of enzymes fits previously with the substrate. So induced fit model, whereas in the enzymes changes shape slightly to fit their substrate. Once a reaction occurs, products are released. Whereas the substrate and enzymes combination, which is bind together and form a reaction, where the products are released into our body. So factors that affecting enzyme activities are the temperature. I have previously explained that optimum temperature. So else your body temperature is also known as an optimum temperature. So too high adenosine enzymes, too low slow reactions may also affect. pH enzymes has an optimum pH. Substrate concentration is more substrate fast reaction, whereas more enzymes fast reactions up to the concentration value of substrate and enzymes. Both the substrate and enzyme concentration value increase the fast reactions. So in enzymes inhibitors. So competitive inhibitors are block active sites. So non competitive inhibitors are change enzyme shapes, can be reversible or irreversible, used in medicine. Whereas we are lagging in enzymes or else in a substrate, whereas these two doesn't bind together and we got sick, it is injected through an external foreign substance, which is also known as an antibodies or drugs. So antibodies is injected to you, whereas you may feel sick or anything else, you have affected by a flu or anything else. So these are all the enzymes inhibitors. So what are all the examples of enzymes? Amylase, protease, lipase and DNA polymerase. So DNA, already you have know about DNA, which is deoxyribonucleic acid. So amylase is break down starch into sugar. And saliva, through our saliva is the major process of digestion, whereas you have chew the food particles in your mouth, and the body's enzymes or substrate which is required for your body is absorbed by your absorbed through the saliva, and the starch into sugar conversion also caused by the amylase. The protease is a protein which is breakdown in the amino acid. So if you are intaking the food which is high in protein, so the protein is a breakdown through the amino acid which is present in your body. So lipase is a fatty acid and glycol. So good fat is also required for our body, whereas we are talking about obesity. So the obese is mainly caused due to carbohydrate and fat molecules, but your body is naturally needs some good fat for the glycerol, so breakdowns of lipase. So DNA polymerase is build DNA starts during replication. So DNA replication, enzyme is a major plays a major role in DNA polymerase. So enzymes in human body is in digestive system, DNA replication and repair, and blood clotting and immunoresponse. So blood clotting is mainly caused due to the coagulating factor present in your body, which may stimulated by the platelets. So platelets is responsible for blood clotting. So else in enzymes plays a major role in blood clotting and immunoresponse, where in metabolism it plays a major role in control energy releasing reaction. So high metabolism rate or low metabolism rate is also dangerous for your human body. So maintaining the metabolism rate is also important in a human body, which is caused by the enzymes. So industrial and medical uses of enzymes are detergents. So enzymes breaks down strains. So every detergents that contains an enzymes. Food industry, cheese making, brewing, juice clarification, or some of the enzymes which are used in food industry. In medicine, we also already we discussed antibodies and diagnostics, drug design treatments, or enzymes replaced therapies. So enzyme degeneration is extreme heat, pH or chemical can denature enzymes. So already we have discussed about pH, whereas in a optimum temperature enzymes works properly. In an abnormal heat or pH value, the enzymes nature will lose and it is denature enzymes. Denatured enzymes equal to loss of structure and function. Denature is often irreversible process. Thank you.